I'm Cassidy. I'm a fourth-year PhD student in the fungi lab at UC Davis with the Department of Wildlife and Fish Conservation Biology and the Animal Behavior Graduate Group. Today I'm going to be sharing with you some of my work investigating the role of feed axis and thermal physiology of Chinook salmon. Let me introduce you to my friend and study species currently swimming around on your screen. Chinook salmon is a species of immense cultural, economic, and conservation importance. Not unlike myself, Chinook salmon is a native California species. California contains a large number of at-risk Pacific salmonids, including this particular fish. Chinook salmon is an anadromous fish species. This means that part of its life cycle is spent in fresh water, while the other part is spent in salt water. This map shows river routes in California, and it is through these rivers that salmon outmigrate. Along this massive migratory route, fish may experience large-scale environmental variation. For this project, I was interested in specifically addressing changes in temperature and food. There's research which has shown that fish with access to more abundant food can thrive in warmer water temperatures. I chose to further focus on thermal tolerance responses. Thermal tolerance can provide ecological insight, and it has the added benefit of providing regulatory insight as well. Regulatory agencies closely manage our river temperatures using planned water releases. Those management practices are informed by physiological parameters like the thermal tolerance of key species. I reared juvenile Chinook salmon under a broad range of temperatures and food availability, both in the laboratory and in a natural field setting. Then, after several weeks under those conditions, I collected measurements on both acute and chronic thermal tolerance. Acute thermal tolerance can be quantified using critical thermal maximum measurements. Our results highlighted a significant effect of rearing temperature. For lab-raised fish in panel A, fish reared in warmer temperatures had a higher thermal tolerance response. However, within each temperature panel, you can see that there was no difference between different feed rates. This trend was further reflected for fish reared in the field, shown in panel B. There were only minimal differences between the field fish, and overall, they performed most similarly to fish which had been reared at similar temperatures in the lab. These trends were further reflected in our chronic thermal tolerance analysis, which we assessed using upper incipient lethal temperature methodology. Again, we saw that the lab fish in panel A were significantly influenced by rearing temperature, but not the amount of feed received. For field fish in panel B, we did not observe differences between field sites. The field fish once again performed most similarly to their lab counterparts reared at similar temperatures. This upper incipient lethal temperature data is especially useful for management applications because we can use it to elicit an estimated LD50 value. An LD50 value is the temperature at which 50% of the population dies. For habitat managers, this can be considered a critical threshold for temperature-related mortality. Thank you for taking the time to listen to my research today. And importantly, I would like to thank the ARCS Foundation for making it possible for me to even get the science off the ground.